Today's lecture is going to be on the generation of antibody diversity, as well as there will be an introduction to immunoglobulin class switching. If you remember from the last class, we have already discussed how every different B cell which is naive has not encountered antigen has already on its cell surface specific receptor, 3/4 of which is identical in all the cells, only 1/4 is hypervariable, and it is this hypervariable region which is at the amino terminus which is responsible for specific interaction with antigenic determinants on the different antigens. We are capable of mounting an immune response which would make antibodies of at least 100 million different specificities. Now, this cartoon shows different B cells, and the recognition of a specific antigen by clone number 4 would lead to activation of this cell, proliferation in the presence of interleukin 4 and 5 secreted by T cells, and then ultimately differentiation to plasma cells. Just to recapitulate your memory, the hypervariable region responsible for antigen binding on the heavy and light chains constitutes any one of the several V gene segments which come in close proximity, in fact join with any one of the J gene segments in case of the light chain gene, and in case of the heavy chain gene it is V, D and J, addition of one more gene segment in the heavy chain. And as I told you before, the heavy chain gene product, which is the heavy chain of immunoglobulin, constitutes or it gives much more regions for binding to the antigen. How is it that only one of the several J segments present and only one of the several V gene segments present combine? Now, this is because of the presence of special recognition signal sequences, which I may not have mentioned earlier. These are conserved through evolution. So, you have two sets of these recognition signal sequences, and it is a rule that recombination or joining would take place only between the two different RSS, one which is one turn RSS. This one turn corresponds to one turn of DNA helix or two turn RSS. Now, this is what would be required in case of the light chain, lambda light chain assembly as well as kappa light chain assembly. Now, since there are 3 segments on the heavy chain, the two RSS rule would need to be modified, and this has been modified in the following way. The same RSS signal in this case which has been depicted, 2 turn RSS are present on 5 prime and 3 prime side of every D region segment, and the corresponding, that is 1 turn RSS, are present on the 3 prime side of every V gene segment and the 5 prime side of every J gene segment. Now, joining between D and J occurs first, and once this combination has taken place, one of the several V gene segments are recruited. Now, the question comes, how is this variability of 100 different molecules to be synthesized, 100 different molecules which have specificity to recognize 100 million different molecules, how are these generated? All the information lies in the genome, but T and B cells, both of them undergo recombination with respect to their receptors, and this is how the generation of diversity is brought about. Now, there are 7 plus 1 different mechanisms that go to providing this process for generating tremendous diversity. First one is multiple germline gene segment, second combinatorial VDJ joining in heavy chain and V and J joining in light chain, combinatorial association of recombined heavy and light chain, junctional flexibility, P region nucleotide addition, N region nucleotide addition, somatic hypermutation. And last but not the least, which does not contribute to the recombination with respect to variability, yet one can say this also provides diversity, because once the VDJ recombination has taken place, then the recombined region can combine with any one of the constant domain regions in the heavy chain, increasing the diversity. That means the same recombined VDJ can associate with M and D simultaneously. It happens on the naive cell as well as any one of the other constant domain gene. Now, let us go to the first mechanism, that is multiple germline gene segment. I have on the slide the estimated V gene segments corresponding to the lambda light chain in mouse, human, the kappa and the heavy chain. Now, I would like to take the simplest one first, lambda light chain. You can see in the mouse there are only 2 variable gene segments corresponding to the lambda light chain in mouse. In human, however, this has evolved to have 100 different V gene segments. Now, when I say different V gene segments, please remember we are referring to the sequence, the nucleotide sequence, which would of course then corresponding to the amino acids. So, now why in the mouse there are only 2 V gene segments? This would mean that the light chain corresponding to lambda is very constricted. It is important to remember here, and I have mentioned this earlier, that 95% of the immunoglobulins in case of mouse are associated with kappa light chain and not lambda light chain gene segment. Now, let us come to the J gene. In human, there are 6 J gene segments on the lambda light chain, 4 in mouse, and now this is something I would like you to see, that though there are 6 constant domain genes corresponding to the lambda light chain in human and 4 constant domain in mouse, all these are identical. Now, kappa light chain, because remember again that light chain would have only one constant domain. Now, kappa light chain, there are 300 different variable gene segments in mouse, 100 in human, 5J in both and 1 constant domain. Heavy chain, interestingly, in the mouse there are 300 to 1000 variable gene segments. Now, that is really a very large number. In case of human, it is 100 V gene segments, but the human heavy chain makes up by having more diversity gene segments. There are 30 in all. Mouse has 13, 6J and 4J in human and mouse respectively, and of course, series of C, that is constant domain gene segments, each one corresponding to the class of immunoglobulin. So, if you have that many different V gene segments, and I have told you joining of VJ or VDJ in heavy chain is a totally random process, and therefore, one can see that this random joining is what gives rise to diversity. And if you do a mathematical calculation, then if you look at just the mouse combinatorial VDJ joining heavy chain, and in case of light chain VJ, then the possible number of combination would happen in the light chain 300 into 13 into 4, that is these three, sorry, the heavy chain 300, 13 and 4, which would give to 1.6 into 10 to the power 4. Now, this along with the light chain, which is 304 because of absence of D segment, would give rise to 1.2 into 10 to the power 3. Combination between these two, combinatorial association, immediately then increases the permutation to an extent where you can have 1.9 into 10 to the power 7, 1.9 times into 10 million of heavy and light chain. So, let us go back and just think that we have now very large number of VDJ and combination between any one. Now, each B cell would have the capacity to join any one. So, randomly you would have any one of J, D and then V being recruited, and then again light chain which is combined separately, V the heavy chain combined separately, and these two coming together after the translation and when folding takes place in the cell, combination of this again would give further diversity. So, therefore, we have covered now the first two, that is several of these gene segments which have the capacity to recombine at the DNA level, followed by combinatorial association between the heavy and the light chain. Let us come to the third, which is junctional flexibility. What is junctional flexibility? Now, since we are talking about two segments which join together. Now, if you remember in the last class, the mechanism of RAG1 and 2 cutting the single strand of DNA specifically at the RSS. Now, RSS, the two are the RSS, which are, if you remember, heptamer and the AT rich nonamer, they come very close in proximity, and this is done by the RAG1 and 2, which recognize these signal sequences, they bind and recognize and they bring them together. Now, cutting of the single strand DNA occurs by RAG1 and 2 precisely at the heptamer. Therefore, though now you get signal joints, that would mean when the heptamer, nonamer, the RSS are deleted, if one sequences these deleted products, one can see that the joining between the heptamer, nonamer, the signal sequences is very precise. It is identical as seen in pre-B cell lines, which are going on to becoming B cells, where recombination is taking place. Now, this is experimental evidence that shows that signal joints are very precise, but when the coding joints join, that it would mean one D with one J or one V with one J or V with DJ. The coding joints always are imprecise, and we will come to how this happens in 2 minutes. Now, I would like you to see this, the coding joints which of 4 different cell lines which are derived from one pre-B cell line. So, this pre-B cell line was now activated to start the recombination process by giving the interleukin 7, as in vivo, where you have the bone marrow providing interleukin 7. Now, the pre-B cells go on to now recombining the heavy and light chain, and when coding joints were sequenced, it was seen the several 1, 2, 3, 4 different cell lines had different sequences. Okay. This imprecise or difference in joining in the coding joint can of course be deleterious, and as seen in the last experiment, the coding joints joining together can also change the sequence, the reading frame in such a manner that you may have the introduction of stop codons, and this is what has happened in the 5 cell lines that were then looked at, other 5, where 2 of them the recombination failed because of the change in the reading frame, stop codons were introduced. However, productive rearrangement happened in 3. Now, this can also, why it is important to know this is because remember when we talked about the development or the ontogeny of B cells, in the antigen independent phase, there are large number of B cells which undergo apoptosis because of lack of precise joining. One of the reasons can be that introduction of one of the reasons is introduction of a stop codon in the reading frame when coding joints were now joined together, changing the reading frame. Next would be, that is 4 and 5. 4, the fourth mechanism in introducing diversity would be addition of P nucleotides, and N nucleotides would be the fifth one. Let us look at the addition of P nucleotide. I have a diagram in the next slide, but let us first go to the theory part. Now, in this particular example, one of the D is joining with one of the J, and by the transesterification reaction, RAG 1 and 2 have made a palindrome sequence. You have now in the coding sequence of the D segment TC and in the lagging strand AG. Now, remember Artemis, the endonuclease, which is specific with respect to recognizing hairpin structures. Now, this protein, this enzyme binds to the hairpin structure and can cleave anywhere, because the cleavage is not specific with respect to the nucleotide sequence. Now, let us see in case Artemis cleaves at the hairpin junction between the D gene segment and this hairpin structure. Now, if the cutting is in the lagging strand, closer to the lagging strand, then you would get TC. Look at the coding strand first. The coding strand would have the TC, which was originally there, plus would get GA from the lagging strand. Similarly, if you look at the J gene segment, and you have now cleavage occurring at the joint after TA, which would mean now that the lagging strand would get here AT from the coding strand. GA, as the color is different color, GA and AT are not present in the sequence of the coding or the lagging strand. They are new nucleotides with respect to that particular strand. When joining takes place, you would have end filling before the ligase activity, and you can see now 4 nucleotides in the coding strand which are different from those that were present earlier. Therefore, now these are known as P nucleotide addition. This, of course, as you can imagine, would generate diversity, but this would also, if the reading frame now adds a stop codon midway, would also be deleterious. However, this is one of the mechanisms of diversity. Let us look at this unique system of addition of N nucleotides. What are N nucleotides? When the hairpin structure has already been cut by the Artemis, as I have already mentioned and described in the P nucleotide addition, there is expression of a specific enzyme, which again is specific to lymphocytes, terminal deoxyribosyl transferase. You might remember from my first slide, my first lecture, that TDT is a marker for B cells and T cells. Terminal deoxyribosyl transferase, immediately you would realize that this enzyme has the capacity to add new nucleotides. So, at least with respect to P nucleotides, the information was available not necessarily in that particular strand, but in the opposite strand. However, in case of N nucleotide addition, these are additions of nucleotides which are catalyzed by the TDT and which are not present at all. Are there any preferences with respect to the addition of any nucleotides? It has been seen that there is no preference as such, but if you look at the number of nucleotides which are present in this particular DJ joining, then there is a preference for G nucleotides to be added. So, now we have two more mechanisms, P nucleotide addition and N nucleotide addition, which would contribute to this diversity. all these are going to change the frame, are going to change the amino acid sequence, which the information of which was available on the coding strand earlier. To look closely at the p nucleotide addition and to make it a little bit clearer, I have a diagram here, the coding strand and the lagging strand, which is in dotted line. Now, it is after all the coding strand that matters. Now, let us look at the hairpin structure. Now, there are two instances here. The Artemis is cutting the hairpin structure at two different regions. Now, in one, if the cleavage occurs closer to the coding strand, okay, then you would have a much smaller nucleotide sequence from the coding strand which is present, and more than half would go automatically to the lagging strand, which I have not shown over here. However, in case of 2, if the cleavage occurs closer on, the cleavage occurs on the hairpin closer to the lagging strand, then you have a much larger fragment which would encompass quite a few nucleotides from the lagging strand, and this would be new to the segment over here, which could be, let us say, D. Cleavage of the hairpin by Artemis is not sequence dependent. Again, I would like to remind you, and position at which the hairpin is cut is variable, leading to variability in the sequence of the coding joint. Let us look at the TDT gene expression. Now, people were able to, when immunologists sequenced these VDJ recombined genes, they found that there were a few nucleotides in the DJ as well as VD, which did not even, they could not count for those on the lagging or the coding strand. And then they realized that there was this enzyme TDT, which was adding new nucleotides. So, after that, large number of sequences were obtained, and it was very clearly seen that in fact, yes, there is this nucleotide addition in the heavy chain gene, N nucleotide addition. I would like to also tell you that N nucleotide addition happens mostly on the heavy chain, because the expression of TDT, terminal deoxyribosyl transferase enzyme, is decreased when recombination of the light chain gene is happening. Now, there is a very elegant experiment that was carried out to show. I have told you in a few lectures ago that people have been able to induce recombination event in fibroblast cells. Now, recombination would not happen, well, let me be precise, sorry. Recombination of the immunoglobulin gene or rearrangement would not happen in any other cell except T and B cells. Well, B cells in case of the immunoglobulin gene, and T cells with respect to the TCR, T cell receptor, which also has, the T cell receptor also has heavy and light chain, etc. The structures might be different, but in any case both of these undergo reorganization. Now, one can induce reorganization of these genes in fibroblasts by expressing a plasmid which codes for RAG1 and 2. So, VDJ recombination was induced in fibroblast cells, and only when these fibroblasts were also transfected with the TDT gene, N nucleotide addition was seen to take place. So, definitely again the relevance of TDT, the enzyme, was very well demonstrated. How many nucleotides can be added to the cut ends? And as many as 15 nucleotides can be added to 3' of both DH-JH as well as VH-DH-JH joints. Now, this constitutes CDR3. This is very important to remember that the CDR3 region is where N nucleotides are added. So, highest diversity would be there. So, N region nucleotide addition, just like to say a few more things with respect to N. There is no template specificity, and TDT adds, like I said a little while ago, DG residues preferentially. Therefore, it has been also observed that VD DJ junctions are G rich. Again, like to mention that end region nucleotides are not added on the light chain, simply because heavy chain assembly takes place first, TDT expression happens at that time, and the level of this enzyme starts going down when the light chain assembly is taking place. During the course of an immune response generated to an antigen, there is not only increase in the titers of the antibodies, but also the affinity. This was looked at a little bit closely. How does this happen? Now, remember this figure. Now, this is experiment that was conducted in a rabbit, injected with one particular antigen on day 1. Serum was, well, blood was collected from the rabbit every other day or every 3rd day, and then the titer of the antibodies generated in circulation to the antigen was determined or quantitated. So, the y axis shows that, the x axis shows the days over which the blood was collected. Now, after the first response, when the titers were seen to come down, the same antigen was injected again and also a 3rd time. Now, there has been slight change, this should be here 60 and this 3 should be here, that would show primary immune response, secondary immune response and tertiary immune response. Now, you can see again, this is what I have told you already, that you would have a much smaller amplitude after the first, which is absolutely expected, and most of these antibodies will be of IgM isotype. You have then memory cells which be generated here, and when the animal sees the antigen again, there is no lag phase. Immediately antibodies, circulating antibodies start appearing, and the amplitude of the response is higher. Similarly, after the 3rd injection of the same antigen, again there is no lag phase and you would see the amplitude which is much higher. Now, when people started to look at the affinity of the antibodies, now this was only with respect to the titer, it was interesting to observe that the affinities of the antibodies increased, and this was every time. In the 3rd immune response, the affinity would probably increase even 100-fold or 1,000-fold. Now, this was then, so people started to look at the receptors on memory cells versus those on the naive cells. Now, we know that the naive cell to a particular antigen already has a predetermined sequence of the hypervariable region. Now, mutations were found in the memory cells after the 2nd and tertiary response. So, there were quite a few experiments that were carried out, one of which I would like to mention over here, because this gave a direct evidence that though the variability or the variable region once formed, that is VDJ, does not alter except for a few mutations which give this the recombination event a better advantage. How? Let us see. Now, there were experiments that were carried out where a hapten. Now, why did these people take a hapten? Hapten is a small molecule. It has an antigenic determinant. It could be either a peptide or it could also be a small molecule such as dinitrophenol. Now, such a small molecule when needs to be conjugated to a carrier protein for it to become an immunogen. In this particular experiment, scientists have taken phenyl oxazolone, which is a very good B cell epitope. You get a good response to this very small molecule, phenyl oxazole. So, it is really, the molecular size is very small. So, this is conjugated and then to a carrier protein, injected into mice, and now hybridoma were established from the spleen of these mice on day 7, day 14 and then subsequently. They got several cell lines, several hybridoma, and now you know what I have not introduced hybridoma, but I would like to just mention hybridoma are those cell lines which are generated by fusing a B cell with a myeloma cell. So, that now the B cell becomes immortal, the antibody producing B cell. Getting a cell line is always easy because several experiments can be done, including looking at the gene sequence. Now, these hybridoma are monoclonal, and therefore, all the cells that are generated from in one particular culture would be monoclonal. So, every sequence would be identical in this particular cell. So, now there are large number of cell lines that were now sequenced with respect to the immunoglobulin heavy as well as the light chain, and as can be seen with the hybridoma that were generated on day 7, neither the heavy nor the light chain differ too much with respect to the immunoglobulin sequence. I would like to say that these small dots and lines represent mutations, okay, and in CDR1, CDR2 and CDR3. Now, I have not discussed these 3 regions, but suffice here to say that both heavy and light chains have 3 hypervariable region even in the variable region, and as the name suggests hypervariable, you have tremendous variability in these 3 regions, and it is through these regions that antigen binding takes place. So, you can see almost no mutations seen in the heavy and light chain in the first 7 days of after immunization. Remember, now the mice were immunized on day 1, 7 days later hybridoma were established. Now, interestingly, when hybridoma were established from spleens of mice that were immunized 14 days prior, which would mean now that this would be the secondary immune response. Okay. You can see very clearly that the light chain has accomplished or accumulated large number of mutations. Now, each one of this, like for example, cell line number 1 has 2 cell, the second cell line has 2, 3 mutations in 3 and so on. Now, this was quite also interesting. Now, this could be only with respect to phenyloxazole, but in the on day 14, it is a CDR1 of light chain seems to have accumulated. In fact, if you look overall, in case of the light chain, CDR1 seems to have maximum number of mutations. Now, the mutations were really quite random. In case of the heavy chain, in the CDR1, 2 as well as 3 regions. Now, in the heavy chain, CDR3 is coded for by part of the day D segment and J. In case of the light chain, CDR3 is only J. You can see the secondary immune response, and you can see in the mutations have increased. You can see by the number of dots. You can see that the mutations are tremendous when you come to the final tertiary. Now, what is secondary and tertiary is day 7 and 14, the animal has been injected here, animal has been injected once more before the secondary immune response and once more and after the tertiary. Now, you can see very clearly that these mutations are accumulated in memory cells in response to the antigen. Now, look at the right side panel. I am sure that it is difficult to read, but I will just tell you what does this show. Now, the antibodies that were synthesized secreted by the hybridoma clones were then assessed for their KD or the affinity to bind the antigen, and what can be seen. Okay, let me read this out. KD into 10 to the power minus 7 moles. So, this becomes 100 nanomolar. So, first the affinity of the antibodies is low, quite low, does not change much in from one cell line to another in hybridoma that were established on day 7. Immediately when hybridoma were established from mice on day 14, you can see immediately an increase in the affinity tenfold, and this became, with this increased further in the secondary as well as the tertiary by 100-fold. Now, this was direct relevance, and this experiment has been reproduced several times to show that during the generation of an immune response, we have increase in the affinity, and this affinity can only happen by receptor editing, because remember the sequence, if it remains the same, there cannot be an increase in the affinity. For the affinity to be increased, there has to be a sequence change, and this sequence change happens because of the presence of an error prone polymerase, and this error prone polymerase gets activated in memory cells. So, therefore, it is only this will of course then the receptor editing as well as increase in affinity can only happen in case of those B cells which are T cell dependent. Remember, memory cell generation itself is a T cell dependent phenomenon. So, affinity maturation, T cell dependence, and then maybe if I have time today, I will talk also about isotype switching. All these happen in T cell dependent B cells. I would like to also tell you a relevant experiment with respect to affinity of antibodies. Now, several years ago it was believed, when people did not know much with regard to these immunoglobulin genes and their affinity, it was believed that if you injected milligram amounts of antibodies, you would get a very robust immune response, but it is not so. In fact, I would also like to say, when one is doing experiments with, let us say, mice and rabbits, rabbit would be, let us say, even 1 kg and a mouse is a paltry 15 grams. The amount of antigen that is injected does not go by the body weight. There is no pharmacological dose. Immunological doses would still be in microgram amounts. Typically, if you are injecting 20 micrograms of an immunogen in mice, in the rabbit you would inject not more than 10-fold, 200 micrograms, but not corresponding to the weight of the animal. Now, there are two instances given over here. Low immunizing dose versus high immunizing dose of the very antigen that I was talking about, and let us say the animals are rabbits. If the animal is injected with low immunizing dose, what would happen? Because the dose is very low, only high affinity receptors of those B cells would recognize the antigen. Let us say this B cell has an affinity of binding to the antigen to the order of 10 to the power minus 11. So, it becomes absolutely to femtomolar range. So, very low immunizing dose, let us say it would be something like 10 microgram in rabbit, you would get an immune response generated, but very few B cells would get activated in response, because only the high affinity ones would. Only these would then get activated, proliferate, differentiate and become plasma cells. Now, maybe in certain instances, you may not detect the presence of antibodies in circulation, because the numbers may be very, very low. Now, the low affinity receptor bearing B cell would not come into the picture at all, because they would not bind. So, there is no question of activation, proliferation and differentiation. Now, if a high immunizing dose is given, then both B cells, those bearing high affinity receptors as well as low affinity receptors, would get activated, differentiate, and you would get a mixture of antibodies which would have both high affinity as well as low affinity. So, effect of immunizing dose on affinity maturation is important, and even if one starts with low dose of high dose of antigen immunization 1, subsequently the dosage can be decreased, so that one would get mostly high affinity binding antibodies. Now, let us go over this whole mechanism of affinity maturation, sorry, not affinity maturation, the establishment of diversity in the immunoglobulin gene. It is because of multiple germline gene segments, combinatorial VDJ joining, combinatorial association of heavy and light chain, junctional flexibility, addition of P region nucleotide, addition of N region nucleotide, and somatic hypermutation. Now, I would like to mention finally that we do have the VDJ, which is already combined, maybe it is undergone mutation, affinity maturation. This can combine, this does combine with always the mu constant domain and simultaneously with the delta constant domain. But those B cells which are T cell dependent can undergo class switching, and in them the recombined VDJ can associate with also any one of the other 5 classes of immunoglobulin, the gamma, alpha, eta. So, I come now, since I have time, I will introduce to you immunoglobulin class switching. Class switching. I haven't introduced again already the 5 different classes of immunoglobulin, but I would have mentioned when I was talking about the genes that you have in the constant domain, you have gene segments that code for constant domain of IgM, that is mu, delta for D. Then there are 4 gene segments for the immunoglobulin G, constant domain G3, constant domain G1, constant domain G2b, and constant domain G2a. Now, this is mouse because in human it is simpler, it is 1, 2, 3, 4. So, human also have 4. Then follows in the immunoglobulin gene organization, you have constant domain eta, that is IgE, and then constant domain alpha. Immunoglobulin class switching is maximum and very high during the naive cell generation. Remember, in the ontogeny of B cell, IgM is the first receptor that gets surface expressed. Now, by alternate splicing, IgD is also simultaneously expressed. So, during this, now though this is not class switching totally from one to the other, there is alternate switching, and this is, like I said again, by alternate mRNA splicing, that you have formation of IgM and IgD on naive cells. Now, class switching is remarkably high in the first instance and remarkably high during the secondary immune response when IgM switches to any one of the classes of immunoglobulin. Classes again would only be referring to the constant domain genes. Isotype switching occurs in the germinal center 1 week after immunization. Now, you have already been familiarized with the secondary lymphoid organs, and you already know what are germinal centers, and this is where you have memory cells, you have T, B and macrophages cells, and importantly memory cells of B cells, which 1 week after the primary immunization, if the antigen still persists, then there is activation of memory cells and there is isotype switching. So, the memory cells which would be IgM. Now, one thing which I missed telling you all earlier, that though we have IgM and IgD, both the immunoglobulins on the naive cell, IgD is almost never expressed, and the only time one sees IgD is in fact in myeloma cell, aberrant. So, there is no IgD in circulation or very low amounts. It is always IgM, IgG, IgE, IgA, mostly IgM and IgG and IgA. Now, 1 week after the primary immunization, isotype switching takes place, and this isotype switching is absolutely requires CD40 receptor CD40 ligand interaction. Again, I would like you to remember that CD40 receptor is present on B cells, constitutively, but require the presence of CD40 ligand, which is made by the T cell for certain processes of activation. It is this interaction which now tells the cells to produce the cytokine receptors and also gives the progression signal to the B cells for proliferation. If there is no CD40 ligand interaction, no class switching takes place, also no memory cell generation, and this has been shown by conditions in human who lack CD40 ligand. Their T cells cannot make CD40 ligand, and therefore, there is no CD40 receptor ligand interaction, therefore, no memory cells generation, no class switching and no affinity maturation, because all this process is totally T cell dependent. Isotype, the same variable, again and again I would like to mention this, that it is already combined VDJ which now can associate or disassociate from the IgM and associate with any one of the isotypes. Forget the IgD, because in fact there is no switch region between these two. Isotype pattern varies according to antigens. Now, I have another slide which goes a little bit more detail, but suffice here to say that, for example, why do I say isotype patterns vary according to antigen? Now, antigens such as viruses usually induce IgG2A type of immune response. Why? Because of the fact that IgG2A, the Fc receptor, the IgG2A can bind to a specific receptor on the T cells, which allow now antibody dependent cellular cytotoxicity. Helminth antigens, they induce IgE type of an immune response or IgG2B. Now, we will be dealing with this a little later. Carbohydrates on the other hand, or cell wall antigens, typically induce IgG3 type of immune response, because, now let me give a reason, IgG3 can cross the placenta, IgG3 can fix complement better than the other isotypes. Now, of course, one can look at it from the point of view that the maternal immune response to bacteria can be conferred to the fetus, and also because IgG3 can fix complement, these bacteria or pathogens can be very well eradicated even utero. Just introduce to you this recombination event that takes place during class switching. This also of course again happens at the level of DNA. So, let us look at recombination. Now, how let us first look at the mechanism. How does this happen? Now, very clearly you can see VDJ has already recombined and fine waiting over here. You have now on the 5 prime side of the constant domain gene segments, which are in different colors, a switch region. There is no switch region, please note here, between the constant domains of mu and delta. Therefore, you can have RNA splicing and alternate expression on the naive cell. Now, switch region mu needs to combine with any one of the switch regions for the isotype switching. How does this happen? Let us say in this particular case, now you remember the organization of the constant domain is exactly the way I have mentioned earlier, and this does not change. So, you have now this cell that is going to undergo class switching wants to switch from mu, that is IgM, to IgG2B. Now, this happens by the coming together of the switch region genes come in close proximity, and then you have the deletion of all the sequences which were in between the M and the IgG2B. There is recombination between the switch region S mu, that is for the mu or the IgM, and gamma 2B, Now, during transcription again you will have RNA and then you have the mature transcript. Along with this you will of course have this interesting structure. This is the part that has been thrown out of the DNA sequence. You have, just like in the other strand that is retained, you have a part of the switch mu and a part of the switch gamma 2b which have joined, and this is part which has been deleted, and you can see the constant domain mu, because now that cell can no more make IgM, neither IgD, neither IgG3 nor IgG1, because all these are in between the switch regions . All these are between the switch regions mu as well as switch region gamma 2b. So, you can just visualize very well that you have the switch regions that come together, they are recognized, come together, then you have cleavage. So, you have a part of the switch region now which is still in this sequence and a part which is here. Now, in the same thing would happen if the switching has to take place between mu to, let us say, IgE or mu to IgA. Now, once let us also then look at something interesting, that if now the cell has already undergone switching from mu to IgG1, then this memory cell can further switch from IgG1 to Ig, let us say immunoglobulin IgG2A, or what is not shown here is immunoglobulin alpha. But of course, since this part would be deleted, the cell can no longer go back to making these isotypes. So, once again there is cleavage and throwing off or deleting an entire sequence. In this case it would be the constant domain genes. Now, I would like to stop here, and in my next class I would like to talk about what brings about this recombination. Is it known? What are the different proteins which bind to this? What induces? How much is known with respect to class switching? What can be these factors that govern? Now, obviously, one can imagine that these factors would be somehow associated with the kind of antigens that go in, because it is the antigens finally which determine which kind of an immune response with respect to the isotype. I have already given you example of viruses which induce IgG2A type in case of mice and IgG2 in case of human, whereas helminths would switch IgM to immunoglobulin E. Obviously, then the immune system has been involved in such a manner that the immune response should help to eradicate that particular foreign object in the body. Therefore, there should be generation of some transcription factor, there should be generation of in turn some proteins by the antigen themselves, the immune response that is seeing this antigen, which would culminate in the production of those kind of immunoglobulins which would be highly effective. So, next class will be looking at the mechanism of this class switching further, and also looking at the immunoglobulin gene regulation. What actually regulates all these processes? Are there any genes which are above RAG1 and 2, and what are these? Thank you.